Okay, so now we'll see DNA replication. Okay, we'll see how one strand of DNA makes another strand of DNA. Okay, so before getting into that, let us start with the basic sequence, the central dogma of molecular biology. What is the central dogma of molecular biology? A DNA produces one more DNA. What is this process called? This is called replication. And then a DNA produces RNA, and this process is transcription. DNA to RNA to protein translation. So this is the central dogma. Now we will see what is happening in this process. How a DNA is producing one more DNA. Okay. Now for knowing that, you should basically understand the structure of a DNA. The structure of a DNA is a double helical structure. We'll see what is a double helical structure. It consists of two strands. A strand means thread-like structure. Okay, so this is a thread-like structure. This is a thread-like structure. This is two strands. This is one DNA molecule. Now these two strands of DNA molecule will be present in this way. It will be present in the form of a helix. This is a helical structure. So that is why DNA is a double helical structure. Is it clear? And there are two terms you should know when we are talking about the central dogma by itself. The first one is autocatalysis. Autocatalysis. Autocatalysis means when one DNA molecule produces another DNA molecule. This is called as autocatalysis. But when a DNA molecule is producing an RNA molecule, then it is called as heterocatalysis. Why it is called as hetero is, from one kind of molecule, you get another kind of molecule. Is it clear? From one kind of molecule, you get another kind of molecule. That is why it is called heterocatalysis. But the first DNA produces one more DNA. So to make you understand, I can tell this is replication and this is transcription. Is it clear, children? So this is replication and this is transcription. We'll be seeing about this. So for knowing what exactly happens in DNA replication, there are a lot of enzymes which play a very crucial role in the process of DNA replication. We'll first see the names of the enzymes and what are the functions they do, and then we'll be seeing about the process itself in detail. You should keep one thing very clearly in mind when we are talking about the enzymes. The enzymes that I've written here are not exactly in the order as it is going to act during replication. These are the enzymes that is going to act. So how they are going to act, when they will act, whether parallelly or when one enzyme stops functioning, the other enzyme is going to act. All those things we'll be seeing in the steps of replication. But overall, these are the groups of enzymes which will play a very crucial role in the process of DNA replication. The first enzyme you should know is helicase. The name of the enzyme is helicase. Enzymes are basically substances which break. Am I right? They break. So you always add a ASC. When something breaks the helix, it calls as an helicase. So this is the helical structure. Am I right? This is the helical structure. This helical structure has to be broken at first. It has to be broken. That means the helical strand has to be brought as two separate strands. So first thing, this helicase enzyme will come. It will act here and it will make sure the two strands are separated from each other. Is it clear? So what it does, it opens or unwinds the helix. It will open or it will unwind. It is like this, no. So it has to be brought like this. It will unwind the helix. And how does it do? It breaks the hydrogen bond in between. It breaks the hydrogen bond in between. How does it break the hydrogen bond? If you remember the structure of the nucleotide, you will be able to understand. It will break the hydrogen bond in between and you will be getting two different strands. So this is the first step in DNA replication. Is it clear, children? Now the second step is, the second enzyme comes which is called topoisomerase. This topoisomerase, what it will do is, see, I am having two strings with me. Okay, I am having two strings with me. And it is coiled up like a DNA. When I remove the two strings from each other, there will be some tension in the string to coil back. Am I right? From wherever I took, it will coil back. So in the same way, if I am using a helicase enzyme and if I am bringing the two strands of DNA separately, there is a chance that tension in the two strands will make them coiled up again. That should not happen. So who will remove this tension? This topoisomerase will remove the tension. The topoisomerase will make sure the tension in the strand is completely gone away. And how does it do this? It cuts the DNA at first. The helicase enzyme cuts the DNA. Cutting means not literally cutting, breaking the hydrogen bond. It cuts the DNA. And then the second step it does is it rotates. After cutting, it rotates. The two strands are rotated. And finally, it releases the strand. After rotating, it releases the strand. They will not come together with each. Is it clear, children? So this is the second enzyme. The third thing is single strand stabilizing protein or enzyme. In short, they call it as SSSP or SSSE. Both the names are there. It is a single strand stabilizing protein or single strand stabilizing enzyme. Now, both of these are very important. What happens is, once the tension is released on both the strands, there is a lot of tension on both the strands. Once that is released completely, this protein come as a final check. They make the DNA stable. If there is any final little bit tension or stress in the strand, that has to be removed completely. And who does that? The single strand stabilizing protein makes sure that there is the DNA is completely stable. It is free of stress. It is free of any kind of tension. Is it clear? The next is ligase. This you would have already come across this name. The function is same. What a ligase does is it joins the DNA fragments. If there are many fragments and pieces of DNA, all these pieces of DNA will be joined together by the enzyme DNA ligase. And when this is going to actually come in the process, when you read it, you will understand. When we see the process, why it is used, we'll get to know. Okay. The next is DNA polymerase 1. There are two kinds of polymerases that is coming in DNA replication. One is DNA polymerase 1. The second is DNA polymerase 3. There are two different polymerases coming. DNA polymerase 1 helps in removal of primer and filling the gap. A primer has to be removed. So what is a primer? We'll come to that. So a primer will be coming and that primer has to be removed. So that is done by DNA polymerase. The next most important enzyme is primase. This primase is the one which helps in the initiation of DNA replication. When I tell initiation, it is the primordial enzyme without which the DNA replication will not happen at all. If this enzyme is not there, DNA replication is not there. Why? We'll see. It first helps in the process of primer formation, primer synthesis. So we'll know what is a primer at first. Everyone knows what is a nucleotide. Correct? Everyone knows what is a nucleotide. It can be a DNA nucleotide or it can be an RNA nucleotide. It can be any nucleotide. If I'm going to tell it's a oligonucleotide, then it means there are a lot of nucleotides that is arranged in the form of a chain. That is an oligonucleotide. This primer is the oligonucleotide of RNA. It is an oligonucleotide of RNA, which means it is a small segment of RNA. It is basically a small segment of RNA, but it is a oligonucleotide. That is the primer. This primer is the first thing that gets attached to the DNA strand during replication. So how does it come? We'll see in the process. So this primase is the enzyme which facilitates the primer synthesis, and this primer is a small segment of RNA. Is it clear? Now go to the next step. This primase is actually called DNA dependent RNA polymerase. It is called DNA dependent RNA polymerase. Why is it called in that way, the primase? The reason is, what kind of primer has to be synthesized? I told you primer is a small segment of RNA. So what segment of RNA has to be produced? What segment of RNA has to actually be synthesized? This will be decided by the DNA itself. If this DNA is going to be replicated, what primer should be synthesized for replication? That will also be decided by the DNA. But helicase is not decided by DNA. Topoisomerase is not decided by DNA. That is there already outside. It is going to come and act. But what kind of primer is going to be synthesized will be decided by the DNA, and DNA itself will produce it. That is why it is called DNA dependent RNA polymerase. Is it clear to you? Very good, children. Now this next one is DNA polymerase 3. Now this DNA polymerase 3 is the main enzyme which actually participates directly in the process of replication. It will participate directly. How it is going to participate? I'll tell you. See, you know about the double-stranded structure, right? So this is the double-stranded helical structure, and this double-stranded helical structure will become like this, correct? So this double structure has to get replicated, which means one strand will be produced this side and one strand will be produced this side. Then these two strands will be coupled together. That is only replication. From the parental strand, two separate strands came. From the two separate strands, two more strands are to be produced. This is replication. Now this DNA polymerase tree will come and sit here. It will come and sit here. So from the parental strand, the new strands that are going to come are going to be passed on completely through the DNA polymerase tree. The DNA polymerase tree is the main enzyme which actually plays a crucial role in replication. It is the one which directly takes part in replication. All the others are accessory, but DNA polymerase tree acts straight. Is it clear? Now the next one, there are three subunits that are basically present in DNA polymerase tree. The first one is called as an alpha subunit. Alpha subunit is a catalytic unit. Can someone tell me what is a catalyst? What is a catalyst? A substance that speeds up or slows down a reaction without actually taking part in the reaction. It will speed up or slow down, but it will not take part in the reaction. So the same thing is done by the alpha subunit. It will make sure the process is smoothly done in a short period of time, because DNA replication has to be fast also, because it is directly involved with cell division. Am I right? So it catalyzes the whole process. The second is the epsilon subunit. Epsilon subunit. See, if I have a code, this strand is replicated, and I have a code like A, T, C, G, G, C, T. If this is the code that is present on the strand, and my replicated strand is having T, A, G, C, and instead of C, say for example, A has come here. It will not come. Just to give you an example, I am telling. So this is wrong. It should not come in this way. If it comes in this way, it means the replication process is by itself wrong. There is some error in DNA copying. There is an error in DNA copying which can lead to mutation, something like that. So who will check this? The epsilon unit from the DNA polymerase check this. They check, they proofread, they make sure whether the formula is correct, the codon is correct or not. Is it clear? Fine. The final thing is the beta 2 subunit. The beta 2 subunit, I told you, DNA polymerase is going to come here. If DNA polymerase is going to come here, and the parental strand is going to go like this, from the parental strand, the replicated strands are going to come. You can find it's just sliding out. It is just sliding out. Am I right? If there is a clamp here, and if you put the parental strand here, the replicating strands will be sliding out like this. So that sliding clamp is actually beta 2 subunit. So to put it more simply, I can tell, for the replicated strands to come out easily without any strain or tension. So whatever job has been done by topoisomerase at first, to release the replicating strands without any tension or stress, this beta 2 subunit will act as a sliding clamp. Clear, children? Now the next enzyme is phosphorylase. This phosphorylase is addition of phosphate. Addition of phosphate is the phosphoric acid, which we saw in the structure of the nucleotide. Is it clear? So that phosphoric acid has to be added then and there to make a nucleoside into a nucleotide. A nucleoside is there. If I keep on adding a phosphate linkage, a phosphoric acid to it, it becomes a nucleotide. And a series of nucleotide will only become one big nucleic acid. Am I right? So this helps in addition of phosphate. Along with addition of phosphate, it activates the nucleotide. Before the process of replication is starting, even before helicase enzyme can act, the nucleotides are to be activated. They'll have to be sensing that replication is going to happen. So that is also done only with the help of phosphorylase. Is it clear, children? Fine. Now the final thing is nuclease. What this nuclease does is, it catalyzes the cleavage of phosphodiester bonds. Where does phosphodiester bonds take place? Where does it take place? It takes place between the phosphate and the pentose sugar. It takes place between the phosphate and the pentose sugar, the fifth carbon of the pentose sugar, and any one free OH group of the phosphoric acid. So if that phosphodiester bond has to take place, and that cleavage, they are not calling it as a direct bond, it's a cleavage. Cleavage means it should stick together completely. There should be no disturbance there. There should be zero tension there. Only then there will be a proper molecule that is formed. Otherwise, the molecule will be highly unstable. So to generate that kind of a molecule, nucleus finally acts in that process. Is it clear? So these are the 9 enzymes that will act in the process of replication. And the most important one of them is primase DNA polymerase 3. These are the 2 main important enzymes which plays a crucial role in DNA replication. Okay.